Cellular respiration, the electron transport chain. Finally, we're going to get some real ATP out of that glucose. So that's what this is all about. You've been wondering why we're getting these dwindling little amounts of ATP along the way. Well, here comes the large amounts of ATP. All right, so remember that where we're beginning here is after glycolysis has happened and the intermediary step where we form acetyl-CoA and the Krebs cycle have happened, and now we're moving on to the electron transport chain. All right, so here is that picture right out of your book in chapter 9, kind of giving you the big picture. So I'm going to remind you that glycolysis takes place in the cytosol, and that's the conversion of glucose to pyruvate in 10 steps. And we have the electron carrier NADH, okay, which is going to be utilized in the electron transport chain, and we get a little bit of ATP out of that. If you remember, we're using 2 ATP and producing 4, so we have a net of 2 ATP from glycolysis. Then pyruvate moves across the membrane via active transport into the mitochondrion and goes through that conversion where a little CO2 is given off and it's combined with coenzyme A to form acetyl-CoA, which gets to go into the Krebs cycle. So for each 1 molecule of glucose, of course, we get 2 pyruvates. So we'll have 2 acetyl-CoAs to go through the Krebs cycle twice. And we get a little ATP out of that, as well as NADH and FADH too. And so all of these electron carriers are going to be utilized in the electron transport chain, where we're going to get a ton of ATP. So that's what's next. And what's the point? The point is to make ATP, and we keep saying that, but here's where we're really going to get the ATP. So accounting so far, glycolysis, 2 ATP. Krebs cycle, 2 ATP. And you probably remember that each Krebs cycle only produces 1 ATP, but since acetyl-CoA, we got 2 of them, the Krebs cycle goes around twice for each molecule of glucose. So for each molecule of glucose, we have 2 ATP from glycolysis and 2 ATP from the Krebs cycle. And okay, life takes a lot more energy to run than that. So we have to extract more than 4 ATP. And we talked about this a little bit in one of the beginning lectures on the ATP. All right, this is I think a pretty awesome thing to think about. A working muscle recycles over 10 million ATPs per second. Okay, so getting 4 ATP out of glucose ain't gonna cut it. So there's got to be a better way, and there is. Ta-da! Electron transport chain. So where does this happen? This happens in the inner mitochondrial membrane. All right, so that's one of the things you need to know about each of the steps, is where it occurs. What part of the membrane, or what part of the mitochondria, what part of the cell do these things happen? So that might be something to make a little flashcard of. Where does glycolysis happen? Where does the Krebs cycle happen? And where does the electron transport chain happen? All right, so this corresponds with the section in your book. The electron transport chain starts on page 170 and goes on to page 172, just so you're following along with your book. Okay, so this occurs along the cristae, those inner folds in the inner membrane. And we're using a bunch of transport proteins and enzymes that these proteins are embedded in the membrane. So that we have the transporter of electrons down the electron transport chain linked to the pumping of hydrogen ions to create a hydrogen ion gradient. We're going to go through this in detail in a second. And we're going to get 36 ATP from 1 glucose. All right, so that's a lot of glucose. Not glucose, that's a lot of ATP out of one glucose. Right, so that's more like it. Right, and this only works in the presence of oxygen. This is all part of aerobic respiration. Okay, remember the anaerobic respiration are the two types of fermentation we talked about, but the Krebs cycle and electron transport chain are part of aerobic respiration. So there's an oxygen. All right, we went over this in the last one, but I'm just going to remind you about the vocabulary here in the anatomy of a mitochondrion. Okay, so we have an inner membrane with the folds that are called cristae. There's so much membrane jammed into this tiny little space because these important chemical reactions are occurring on the membrane. All right, and also there's significance to the intermembrane space, these fluid-filled spaces between the membranes. Okay, so in the inner membrane is where the electron transport chain takes place. So this arrow is leading you to show you, okay, that if we enlarge this inner membrane, this is where the electron transport chain is. And you can see it looks kind of like other membranes we've talked about. It's a phospholipid bilayer with embedded proteins, integral proteins. All right, so that's just showing you that we are in the inner membrane. Okay, and there's the intermembrane space, so that when we're talking about transport across, we're not talking about from the cytosol, we're talking about from the intermembrane space of a mitochondrion. Okay, and in here is the mitochondrial matrix. All right, so this is the first three major complexes we're going to talk about. Let me pull up all the images here so it'll be easier. This picture goes along with the picture on page 172, where we look at a little bit of the details. Oops. Okay, so we're going to go through each part of these steps, all right, but this just gives you the gist of how electrons are moving through these three complexes, okay, and each of these complexes is just an integral membrane protein, okay, and these arrows out are the H pluses that are going to be pumped out as the electron moves on. So the electrons move along, hydrogen ions, protons get pushed out, and that continues through these three complexes until there's just electrons, but we create a concentration gradient because we end up with a bunch of protons out here. And we'll go through that. I'm saying this rather quickly, but this is just to give you an introduction to what the whole thing looks like that we're going to be talking about, because we're going to be NADH and FADH2 have been carrying electrons for us, and they're going to ditch their electrons in the electron transport chain. And the electron transport chain is going to transport them along, and while that happens, we create a change in the gradient of hydrogen ions or protons, because they're just little H pluses. And then once we have a significant difference between the outside and the inside, and we have establish a concentration gradient, then those little H pluses get to power our ATP synthase. All right, so what's the big picture of the electron transport chain part of this is that we're using our electron carriers so that we can move our electrons along to power the creation of a concentration gradient of protons. Okay, and that sentence will make more sense in a second. Well, maybe a full minute, because we're going to go through it piece by piece. This is just to show you where it is happening. You still with me? I hope so. I may have said that all in one big babble. Hope not. Okay. Okay, remember the electron carriers. All right, so I'm going to remind you about where our wonderful, powerful electron carriers came from. So during glycolysis, okay, we got 2 NADH. Okay, so from one molecule of glucose during glycolysis, we got 2 NADH. Then during the Krebs cycle, okay, we get 8 NADH and 2 FADH2s. Okay, now the reason, if you notice here in the picture at the Krebs cycle, we have 3 NADHs here and 1 over here. So that's just 4. So why did we get to 8? Well, remember that pyruvate was just half a glucose. And so for one molecule of glucose, we're going to go through the Krebs cycle twice. So that's where the 8 comes from. Similarly, from one turn of the Krebs cycle, we get 1 FADH2. But because we go through the whole thing twice, we end up with 2. So tallying that up, we've going into the electron transport chain, we have 10 NADH and 2 FADH2s.
Oink, oink.
And we've been showing them as little piggy banks because they're hanging on to these high energy electrons for us so that they can carry them someplace else. Okay? So the whole point of these molecules, like what do we care about NADH and FADH2, is just that they carry electrons. Right? And so now it's time to use the energy that they're holding in these high energy electrons that we got from food. Okay, so the first step is to, for NADH to break down into NAD plus a hydrogen. All right, so that's what's happening in this first step with NADH dehydrogenase. And you can tell by the name dehydrogenase, what is it doing? It's dehydrogenating this molecule. Okay, and there's our high energy electron. Okay, so with the oxidation reaction that you're looking at here, where NADH becomes NAD plus a hydrogen, okay, that's going to happen in this first step. There we go. This hydrogen, all right, is going to give us our electron. All right, we're going to be able to use that high energy electron. So this hydrogen, what we're getting out of it is the electron and a proton. Okay, so it's a hydrogen ion, but a positive hydrogen ion is just a proton. So we can use those terms sort of interchangeably here. So I may say hydrogen ion, I might say protons, but you know what I'm talking about. All right, and these are kind of the key for what we're getting from having a high energy electron that's going to drive us to make some ATP. Right? So the whole point of this last part of cellular respiration, these 3 complexes, is to move along high energy electrons and at the same time be moving protons into the intermembrane space so that we create a concentration gradient. Okay? Protons are not going to diffuse back across the membrane because, of course, they have a charge to them and they're polar, and polar things can't go through the membranes. Okay? So to reiterate again, okay, we're using our NADH to get an NAD plus and an electron and a proton. The electrons move along the complexes, as you'll see in a second, all to create a proton concentration gradient so that hydrogen ions are being pumped out into the intermembrane space. And we're going to use those to make ATP later. Okay, so here is that reaction, NADH to NAD+, and then we've got a bunch of NAD+ hanging out in the matrix to be used elsewhere to become NADH again. Okay, so it's a recyclable molecule, remember. Okay, then, all right, so here's our hydrogen, all right, which splits, and there's our electron, and out goes a proton, right? Then, okay, and the electron gets carried over, okay, out goes another proton, all right, here's another electron, and out goes a proton. All right, and here's our FADH2, which is going to do something similar here and give off that hydrogen, all right, to give an electron and a proton. All right, NADH is going to lead to a little bit more energy than FADH2 does. Okay, this is a little bit higher energy. Right, and all right, we're also going to have 2 hydrogen and 1 molecule of oxygen to give us a byproduct of the water right here. Right, and what was the whole point of this? Well, the whole point was to build a proton gradient. Okay, that's the big picture. Why do these carriers go through the electron transport chain? To create a proton gradient. That's why. Okay, what are we going to use that proton gradient for? Well, that's what we're going to use to make ATP. So you need to understand the big picture of this. Don't obsess about the names of every molecule, but understand that our electron carriers have carried high energy electrons to the electron transport chain in the inner mitochondrial membrane. Electrons are being used to create a proton gradient. That's the big picture. Okay, so what powers the proton pumps? All right. So stripping hydrogen from electron carriers, just to go over again how that occurs. Remember that oxidation refers to the loss of electrons to any electron acceptor, not just oxygen. Okay. Hydrogen is cleaved off of NADH and FADH2. We just saw that happening in here as I was showing you where those electrons were coming from. Okay, the electrons are stripped from the hydrogen atoms, okay, to give us electrons and protons as 2 different things. Electrons pass from one electron carrier to the next in the mitochondrial membrane, okay, and that's electron transport chain, not etc. So flowing electrons is energy to do work. Okay, these are high energy electrons driving a process. All right, and the whole point is to create a proton gradient. Okay, transport proteins in membrane pump the protons across the intermembrane space, across the inner membrane to the intermembrane space. All right, so this is what you have to know about the electron transport chain. Okay, this is giving you the big picture. So you might want to pause here and jot these down if you didn't print out the PowerPoint ahead of time. But this is the essence of this process. All right, we're getting energy to drive this process from the electrons. And why are the electrons important? Well, because we're going to create a very, very necessary concentration gradient of protons. Right? So right now, the concentration gradient is that we have a higher proton concentration outside than inside the inner membrane. Okay, and that concentration gradient, we've kind of hinted at it a bunch of times, but now we're putting this big picture together. That concentration gradient is driving this enzyme. Okay, do you remember what this enzyme is? Right, it's ATP synthase. Okay, so it's like a little machine. All right, and this is, you know, these are relatively new discoveries. There's still a lot of things not understood about the electron transport chain. Okay, but the discovery of ATP synthase, which occurs in many membranes, not just this membrane, all right, is how we go from ADP with a phosphate to be combined into ATP. Because remember that the phosphate groups have a negative charge to them, so it takes energy to push them together, and the energy for that is coming from the concentration gradient of protons. So having a high concentration of protons on the outside triggers the protons to move through this little machine. This little machine starts to spin, and we get ATP. So I hope this makes sense. All right, there's that ATP. Ta-da! Moving electrons do all the work. All right, but what pulls the electrons down the transport chain? How are they pulling along here? Okay. All right, we don't want to use ATP. Okay. So what's pulling it, okay, is oxygen. So oxygen is two oxygen atoms that are looking for electrons, okay, and they're going to be pulling the electrons through the transport chain. Right, so the answer to this question is oxygen. What pulls the electrons down the chain? Oxygen. Right, so these three complexes together, creating the proton gradient that we talked about, that's the electron transport chain. This last step of ATP synthase is called oxidative phosphorylation, and we already should know that term from before. But so these are the two parts that go together for the end of cell respiration. Okay, and this in your book we call chemiosmosis. Okay, so those terms can get a little confusing. So your book says that the whole process altogether is called oxidative phosphorylation, but this PowerPoint is a little bit different. So I'll have to check. So your book says that this part is the electron transport chain, this part is chemiosmosis, and together it's oxidative phosphorylation. So I will check on that. All right, so there's the oxygen that we were talking about. I showed you that reaction at the end there, but I didn't want to go into it until we were here so that you could see, okay, this reaction of two protons and one oxygen, okay, forming water. So the oxygen pulls the electron transport chain. Okay, electrons flow downhill to oxygen. Okay, oxidative phosphorylation. Good? Okay, so that part is, I think, in your book as well. I'm checking pages here. Okay, the oxygen pulling part is what figure 9.13 is all about on page 171, if you want to go over that. So it's page 171, and I think we're going to talk about it in the next couple slides. Yeah. All right, so electrons are flowing downhill in the electron transport chain. Electrons move from molecule to molecule until they combine with oxygen and hydrogen ions to form water. Okay? Electrons move in steps from carrier to carrier downhill to the oxygen. So this flow is what's driving the electron transport chain. It's the electrons being drawn to oxygen. Okay. All right, so the reason they're showing you these steps here is because this is not really, this is not costing us energy. We don't have to invest ATP in this process. It's just moving down a bunch of steps from high energy to low. Okay? So since the electrons already have a bunch of energy in them from food because we broke all those bonds, okay, it's got a bunch of energy and it's going to flow downhill toward the oxygen. Okay, each carrier along those three steps, the three carriers, is a little bit more electronegative. So it's pulling a little bit more, and it's controlled oxidation. It keeps it in the steps. All right, so that we have a controlled release of energy so that we can create that proton gradient and power ATP synthase so that we can make ATP and not fire, right? We did it, okay? We set up a proton gradient, hydrogen ion gradient. So the protons flow through ATP synthase, which is like a little machine in an enzyme, so that we synthesize ATP. And remember that reaction? All right, ADP plus a phosphate yields the ATP. Are we there yet? There we go. Okay, it's called a proton motive force. That's a key vocab term. It's also in your book. So that's one you do have to know. All right, so the chemiosmosis is the diffusion of ions across a membrane. Okay, it relates to the generation of ATP by the movement of hydrogen ions. All right, so this is more consistent with what your book says, that's what's happening in that with the ATP synthase. Okay, so chemiosmosis, the diffusion of ions across a membrane, building up a proton gradient so that protons can flow through ATP synthase to build ATP, and it's passive transport because we're just going from high concentration to low. All right, and it's called, you see the word, the root in there, that osmosis is there, and the reason for that is the guy who proposed this thought it was kind of similar to osmosis because it's the diffusion across a membrane using like a channel protein. So he thought it was kind of similar, but it's chemical instead of water, so that's why it has that name. Okay, so chemiosmosis links the electron transport chain to ATP synthesis, and that's, of course, as the penguin says, that's the whole point, because we're trying to get energy out of food. All right, this is the scientist. As you can see, this is kind of relatively recent stuff. We've been building on these ideas for a long time. Okay, let's put this whole bad boy together. All right, so here's the pyruvate that we got from glycolysis. Goes across, remember there's that intermediary step where we form acetyl-CoA. We give off a little carbon dioxide and an NADH. Okay, then we go through the Krebs cycle where we get 1 ATP, some more CO2, some NADH and FADH2. All right, and then so all these electron carriers, the electrons are harvested and carried to the transport system. Okay, because remember the Krebs cycle takes place in the matrix, and the electron transport chain is in the membrane. So number 2, electrons provide energy to pump protons across the membrane. So the electron transport chain pushes hydrogen ions out into the intermembrane space. Oxygen joins with protons to form water. Remember, oxygen is the driving force of the electron transport chain, pulling the electrons along. Okay, and then protons diffuse back down the concentration gradient, driving the synthesis of ATP. Good so far? Okay. All right, and then we produce massive amounts of ATP. So let's look at it all together. All right, so now we can account for our ATP that we're going to get along the way. This is that same picture again and again. So for glycolysis, we're netting ATP. Okay. Plus 2 ATP from the Krebs cycle. Okay. Plus about 36 ATP from the electron transport chain and oxidative phosphorylation. So all in all, from one molecule of glucose, we get around 40 ATP. And the reason it's approximate is because it doesn't always come out exactly the same. Somewhere I saw how many ATP you get for each NADH. I think for each NADH you get approximately 3 ATP, and for each FADH2 you get 2. So that's about where it comes from. So we're creating that giant proton gradient, and then it produces lots of ATP because it powers the ATP synthase. Okay, so that's a lot of work to go from taking sugar inside a cell to getting a bunch of energy, but the cell, as complex as it is, cells are very efficient at this, especially your skeletal muscle cells. They're doing this no problem with a ton of mitochondria in each cell. Okay, summary of cell respiration. So here's our overall equation, right? Where did the glucose come from? Well, we ate it. Where did the oxygen come from? We breathed it in. Okay. Where did the carbon dioxide come from? Well, oxidized carbons get cleaved off of the sugars in the Krebs cycle. Okay, so some of those, when we break some of those bonds, we give off some carbon dioxide. So there's carbon dioxide given off in the Krebs cycle and right at that intermediary step when we form acetyl-CoA. Where did the CO2 go? Well, we exhaled it. Where did the water come from? Well, it came from oxygen after it accepts the electrons in the electron transport chain. Okay, that's why oxygen is necessary for cellular respiration. Okay, where did the ATP come from? Well, we got a little bit from glycolysis, we got a little bit from the Krebs cycle, and we got a ton of it from the electron transport chain. Okay, and what else is produced that is not listed in this equation? Well, we didn't list our electron carriers, which are so important, NAD and FAD, plus some heat that is given off in this process too. And here's the big question: why do we breathe? Okay, so you thought before this that the reason for breathing was so that you get oxygen in your blood. Well, then why do we get oxygen in our blood? Well, so that the blood vessels can carry oxygen to every cell in your body. Why does every cell in your body need oxygen? It needs it to do cellular respiration. So that's why cellular respiration and breathing are both called respiration, because we're exchanging oxygen and CO2. Okay, so the whole reason for breathing is to get oxygen to the mitochondria. All right, what is the final electron acceptor in the electron transport chain? Oxygen, okay? So what happens if no oxygen is available? The electron transport chain is going to back up, and there's nothing to pull the electrons down the chain. And NADH and FADH2 can't unload their hydrogens, and you stop making energy. Okay? That's why oxygen is crucial for this reaction. Okay? So without oxygen, you die. All right, so I think we're showing you cyanide because that's what cyanide does. Yeah, because cyanide's going to stop at the final step here, and then no ATP is produced, and that's why cyanide's a poison. Okay, what's the point? The point is to make ATP. Ta-da! And that's it for the electron transport chain. All right, hopefully tomorrow I'll put together a big review to help you go over all of this all together.